Experiment in this lab, starch storage in leaves, was intended to help us understand what plants do with excess amounts of glucose that they produce during photosynthesis. And what they do with it is they form glycosidic linkages, and they eventually form molecules of starch, which are stored in their leaves, as the title implies. So the way that this was set up was we had two coleus leaves in different situations. One coleus leaf was exposed to light for an entire week. So here's my leaf. The other coleus leaf was not exposed to light. After the week-long incubation in either light or no light, these two leaves were then boiled in ethanol. Okay, so the purpose of boiling them in ethanol was to remove the pigments from these leaves. Okay, because we don't want these leaves to be green when we're trying to figure out if they have starch in them. We need to remove the pigments. So we boil them in ethanol, that removes the pigments. The next thing we do after we boil them in ethanol is we place them in water. Okay? That water rehydrates the leaves because boiling them in ethanol actually makes them very, very brittle. So then we place them in water, and having removed all the pigments from these leaves are no longer green, they are at this point white. So when we rehydrate them, we've got white leaves that we're rehydrating. So now we've got these rehydrated leaves that have no pigment in them, and we want to detect starch. So which reagent are we going to use to detect the presence of starch in either one of these leaves? So we're going to use iodine. Okay, so again, light, no light. Boil in ethanol to remove the pigments. We have white leaves at this point. Rehydrate with water, then add iodine to determine if the leaf does or doesn't contain starch. And what we found at the very end of this entire experiment was that if the leaf was exposed to sunlight, it was indeed performing photosynthesis. If it was not, then it couldn't perform the light-dependent reactions and had to instead tap into its reserve supplies of starch. So knowing that, which of these two leaves do you think is going to have excess amounts of starch in it? The one that had light provided or the one without? And what we saw at the end of this was that the one that was exposed to light had starch, which of course was denoted by a blackish color. The one without light was just turned a brownish color by the iodine. That means no starch. Alright, so that's all that you really need to know from all three of the experiments during the photosynthesis lab. The next lab, of course, is cellular division. I will not be doing a review on that. Instead, I will simply be posting videos that are associated with that particular topic, because I believe they can probably explain it just as well, if not better than I can, and the fact that they'll have better animations rather than me drawing will probably make it easier to understand. So the next unit I will do after this will be biotechnology.